Deoxyribonucleic acid, better known as DNA, is the most fundamental part of a cell. The DNA, which lies in the nucleus of a cell, governs all of the cell's activities, from producing proteins to digesting food, and determining your traits, from hair color to whether or not you will need glasses. Deoxyribonucleic acid takes the physical structure of a twisted ladder-like shape, also commonly known as double helix. DNA belongs in the nucleic acid group of macromolecules. This means that all DNA molecules are made up of nucleotides, which are made up of three parts: sugar, phosphate, and a nitrogen base. While deoxyribose makes up the sugar component, DNA nucleotides can consist of four different nitrogenous bases. These bases are adenine, thymine, guanine, and cytosine, and they're what makes up your unique DNA code. Furthermore, these bases come in complementary pairs: adenine and thymine, then guanine and cytosine. Since they are complementary pairs, in a single strand of DNA, they can only form hydrogen bonds with each other. Adenine bonds with thymine, and cytosine bonds with guanine. These bases form two groups. Adenine and guanine are called purines, and cytosine and thymine are called pyrimidines. These are classifications of nitrogen bases and can be identified by the number of carbon-nitrogen rings they have. Pyrimidines have one ring, while purines have two. Now that we have reviewed the structure and components of DNA, let's dive into how DNA replicates. This process is necessary for both mitosis and meiosis, so that the daughter cells have their own set of DNA. It begins with the enzyme helicase, which unzips the DNA strands of the hydrogen bonds between the nucleotides. To prevent the separated strands from coiling back together, single-strand binding proteins and the enzyme topoisomerase must be present and active. This turns the DNA into two strands, which become part of the two new DNA molecules being formed. Now the enzyme DNA polymerase comes in. This enzyme replicates the separated strands of DNA and simultaneously makes sure that the order of nitrogen bases is the same in the new strand. Also, the enzyme primase must show the DNA polymerase where to start. If this process is messed up, dangerous mutations can and will occur. Finally, the enzyme ligase must glue the new and old strands back together, creating the finished product, double the DNA. To be more specific, this process is known as semi-conservative DNA replication, and it occurs in your cells about 2 trillion times per day. With this in mind, it is imperative that this process is always consistent and accurate. After DNA replication and subsequent mitosis or meiosis, the DNA must somehow be stored in the nucleus. In a human cell, the nucleus contains about 6 feet of DNA. For the DNA to fit properly, several levels of packaging must be used. First, the individual DNA double helixes are wrapped around histones, round structures used to organize DNA. Next, chromatin strands consist of multiple histones. Chromosomes are the final level of packaging, where chromatin strands form larger X-shaped structures. This packaging is essential for DNA to fit and function properly inside of the nucleus of cells. In conclusion, the structure, packaging methods, and replication process of DNA ensures that we can function properly and without 7 eyes on a daily basis. Nucleotides give DNA the ability to be interpreted by the cell. Also, the packaging of the DNA in the nucleus allows it to fit inside our cells. Finally, three special enzymes, four if you consider it: helicase, DNA polymerase, primase, and ligase, ensure that DNA is properly replicated. Thank you for watching and enjoying our video.